So most of the time people think about elimination of the drug as an activity of CYP450. So this is a kind of a diagram to show you that how an individual's variation in cytochrome P450 can affect their ability to metabolize drugs. So we have a bunch of people here, some are light blue, some are dark blue. That indicates the variation of the gene. Over here, the dark blue people, they have active forms of cytochrome P450, so they're able to break down the medicine and clear it out of the body, so you don't get any buildup of the medication, and there's no risk of overdosing on the medication. But the light blue people may have variation that results in a less active form of the enzyme. And so they are not able to break down the medication and clear it out of the body the same. And so they are exposed to a larger dose of the medicine because they have a medication buildup. And they can have an overdose type experience in some situations. So on this slide, I want to show you how you can kind of predict what kind of phenotype a person will have, whether or not they have an active form of cytochrome P450 or not. And also introduce you to some of the nomenclature that we use when we talk about metabolizer status. So in the first row, we have a person with no variant alleles. They have two green copies of the gene. Here those mean wild type, or typically with cytochrome P450s, you're talking about star 1, star 1. Those are the wild type form of the gene. So two copies of the star 1 allele. So that means they have 100% wild type. And their predicted phenotype is called an EM, or extensive metabolizer. Extensive metabolizer is a term that when you hear it, you can think of normal metabolizer. It means the majority of the population is able to metabolize drugs at this particular rate. If you have one copy of the star 1, but one copy of a variant allele that either encodes for an enzyme that has no function or decreased function, your overall enzyme function that's produced in your body is reduced. So if you have one wild type copy and one copy of the gene that encodes an enzyme with no function, you're looking at having about 50% of the functioning enzyme in your cells compared to the person that's star 1, star 1. This is referred to as an intermediate metabolizer, or an IM. You're able to metabolize the drug, just not at the same rate as somebody who is a star 1, star 1. In a person who has two variant alleles, two alleles that either encode for enzyme with no activity or reduced activity, then the enzyme function is greatly reduced in that person, and that person is considered a poor metabolizer, or PM. You'll hear these terms over and over again when dealing with any kind of pharmacogenomics with cytochrome P450s, the EMs, IMs, and PMs. Remember, an extensive metabolizer just means normal. In some cases, especially for CYP2D6, there are situations where you can have more than one copy of the allele on a chromosome. It's a gene duplication event. In this case, you have more functioning enzyme than the star 1 normal metabolizer person, because you've got 3 copies of the gene making the enzyme. This is called ultra-rapid metabolizer. This person is called a UM, and they metabolize drugs much more quickly. I just want to note here that the way the star alleles are typically defined is by testing for the variants used to define specific star alleles. So what I mean by that is a test will test for specific star alleles, say star 2, star 3. If those variants, remember from the grid, if those variants that are present in the star 2 and the star 3 are not found in a person, then the test defaults the star allele assignment to star 1. So that's one of the issues with a test that's not sequencing, doesn't test for all the positions. If you're testing for a limited number of star alleles and you don't find that variation, then tests default the subject to star 1. That means that the star 1, this is really important, extremely important to note, because it means in any situation where you have a star 1 allele assigned, it means that the variation tested for was not found. So in theory, a star 1 is a wild type, normal function. But what I need you to understand is in reality, sometimes people are mislabeled as star 1 just because the particular test that was used to define their star alleles didn't pick up the specific variation they were testing for. So in the case of testing only for star 2, star 3, they could be a star 6, a star 8, and you wouldn't necessarily know. You would just get a star 1 result. Unless you're sequencing, you can't be 100% sure that your star 1 is really a wild type. And this impacts metabolizer and functional groupings, as you can imagine. And it can present a problem when grouping people for study. In research studies, they often group people by metabolizer status. They genotype them, group them together by their assigned metabolizer status. And when you're comparing poor metabolizers or ultra-rapid metabolizers to the wild type, it's important to know that that wild type group could have some variation in it, and sometimes it can confound the results of your experiment.